A very good evening to everyone. Okay, let's start our today's class. But before starting the class, just let me know whether I'm audible and visible to you or not properly. Okay, here is the technical poll. Please let me know I'm audible and visible to you or not. Okay, great. So let's start our today's topic. So as we have already started this topic yesterday, and we have started doing the uses of microbes in the household products, in the industrial product, and we have also studied the uses of microbes in the sewage treatment, right? So today we are going to start with the microbes, the use of microbes in the production of biogas. As you know, we have a little discussed about the biogas yesterday, that biogas is produced by the decomposition, by the anaerobic decomposition of the organic matter, right? It is formed by the anaerobic decomposition of the organic matter, which is obtained from the animal excrete, right? Now we are going to study it in detail. So as you know, the biogas which we have discussed yesterday, biogas is a mixture of three different gases. It is a mixture of three different gases, that is CH4, that is methane, H2S, hydrogen sulfide, and CO2, right? So the three major gases which evolve during the degradation or the decomposition of the organic matter is CH4, H2S, and CO2. But out of these three gases, the main composition or the main component in the biogas is CH4, that is methane. So here it is also written, the biogas is a mixture of gases, mainly CH4, and the bacteria which is used to produce the biogas are anaerobic in nature, and they are usually found in the gut, right? They are found in the gut of cattle. It is found in the gut of cattle, and the microorganism, or we can say the bacteria, which is found in the gut of cattle is methanogen. They are methanogens. So these methanogens, when grow anaerobically and start decomposing the cellulose material. Why the cellulose material? As you know, the cattle, the basic food the cattle are having is the plants, the grasses, right? So as you know, the plants and the grasses have the cell wall of cellulose. So these methanogens are decomposing that cellulosic material, and while this decomposition, they are producing methane along with carbon dioxide and hydrogen. Okay, and the example of such methanogen is methanobacterium. The example is methanobacterium, right? So as I said, this methanobacterium is found in the anaerobic sludge and the rumen of the cattle, that is the gut of the cattle, so that it helps in the digestion of this cellulosic material, right? So this is the diagram of the biogas plants. As you can see here, we have the three different tanks of three different sizes. The first tank is the all these tanks are made up of concrete, right? It is a concrete tank, right? Okay, so what happens in the first tank? We put the organic material, we put the organic matter in the form of sludge or the slurry, right? Here we making the slurry of the organic matter, and then we put it in this chamber, right? Through this chamber, as you can see, this chamber is connected to a larger tank. It is connected to the larger tank, which is known as a digester. It is known as digester. This larger tank or the digester is completely packed. It is throughoutly completely packed so that the no interference of gas can take place. So as the slurry of the organic matter reaches this digester tank, the condition here present is already anaerobic. It is also present here is anaerobic, and due to the anaerobic condition, the methanogens start their working, and when they start decomposing the organic matter, they started producing the methane and carbon dioxide gas. And now you can see in this digestion, in this digester, we have a chamber at the upper portion. This chamber is also known as a gas holder or gas chamber, where the gas which is evolved during the decomposition of organic matter get collected. And to remove this cat or to extract out this gas, we have a pipe, right? We have a pipe which is known as the gas outlet, through which the gas is transferred to the different region or the different houses. Okay, it is this pipe is used for the supply of this biogas to the different locations. Now we are moving towards the the solid which is formed after the decomposition, right? As the decomposition takes place, it started fermenting kind of thing. It started rising in level. So whatever amount or the whatever organic matter get get digested, it start moving towards this next chamber through this outlet. Now the sludge which is formed here after the decomposition by the methanogens, they are used as a manure. They are used as a manure, or we can say an organic fertilizer, which provide a good nutrient medium for our plant to grow. Okay, so what we have done, we have made a biogas, right? And this biogas production technology is developed by the combination of two institutes. First is the Indian Agricultural Research Institute, that is IARI, which is present in Pusa Road, India, Delhi. And another is Khadi and Village Industrial Commission. They both get together and they develop this technology of the biogas production so that we can reduce the use of chemicals, and we can also get the alternative form or alternative fuel for the LPG. Okay, as you know, the fossil fuels are day by day declining, right? Their levels are declining, so we have uses. Now, so here is a question for depending on this biogas. So your question says, what do we collectively call the biogas producing bacteria? What are these bacteria known as who are helping in the production of the biogas? Your option A, the archaebacteria, option B, the cyanobacteria, option C, the eubacteria, and option D is methanogen, right? I'm again repeating it once again, and while repeating it, I'm launching the poll, so you can give your answer through this poll. So option A is archaebacteria, option B cyanobacteria, option C eubacteria, and option D methanogens. Okay, so you have to give your answer. Okay, as everyone has given the answer, so you have given the answer methanogens, right? Because we have just studied these bacteria are able to decompose the organic matter in the absence of air, or we can say in anaerobic conditions. Okay, so it is clear to you. Now we are moving to the next application of the microbes, that is they are using as the biocontrol agent. They are using as a biocontrol agent. Okay, let's study the first term. What the meaning of biocontrol? As you know, the bio term is related to the living, right? And control is means something is inhibiting, right? We are inhibiting something, right? So when we are talking as the biocontrol agent, that means these microbes are acting to control the growth of some biological organism. right? So we can say it biocontrol is the uses of biological method, and in this biological method we are using what? We are using microorganism for controlling the plant diseases and pests, so that the plant, the crop will not get destroyed, right? And for specifically we are using the ladybugs, which control aphids, and dragonflies, which control mosquito, because it feeds on the larva of the mosquito, right? So as you know, the chemical pesticides and insecticide, whatever chemical we are using, either it is a pesticide, insecticide, or even a fertilizer, they always remain very harmful, right? Because they usually get mixed with water, and they also get absorbed by the soil. They get mixed in some nearby water resource, so due to which it leads to the human as well. It leaches to the another organism, other animals, and humans itself. So as you know, they are the chemical, and these are the harmful chemicals which are already inhibiting the growth of some another living organism. Say also uh, play a very harmful role. They are making a very harmful effect on the humans as well. Okay, so that is why we are now shifting towards the biocontrol agent, right? Because by using the living organism against the living organism, we are not using any harmful chemical. And when we are not using any harmful chemical, they are not going to harm us. Okay, so we can say the chemical pesticides and the insecticides kill both useful and harmful organisms and cause pollution as well. And biocontrol method has no such problems, because they are already insects which are present in our atmosphere. We are not introducing any foreign species or something like that, or we are not using some harmful chemical. Okay, so first, our biocontrol, microbial biocontrol agent is Bacillus thuringiensis, BT. You are very aware of this BT term. We have done in biotechnology. BT cotton was there, BT maize was there, BT we can say the BT maize was there, right? So there were several BT vegetables are present, several BT crops were there. So they basically control the butterfly caterpillars. Okay, we have studied there. They were controlling the cotton bollworm and corn borer, right? Along with that, they also control the butterfly caterpillars. As you know, whenever the butterfly caterpillars are present there, they start eating the leaves and the stems or the branches of the plant so frequently, because they are so hungry that in within a day they can digest even half of the plant, right? So the dried spores of the BT are mixed with water and sprayed on the vulnerable plants. The vulnerable plants are those which are affected, right? Which are affected by the pathogen or some insect, such as brassica, that is mustard, and the fruit. Okay, these are eaten by the caterpillar. They are very tasty in, yeah, you know, can say they are the very tasty food for the caterpillar, so they usually enjoy it. So in their gut, the BT toxin is released and the larva get killed. We have studied about this. The Bacillus thuringiensis have a gene that is known as cry gene, and that cry gene is making a toxin that is known as BT toxin, which is initially a protoxin, that is inactive in nature. But when it reaches to the gut of the insect, on reaching the gut of the insect, it gets the alkaline pH. It gets the alkaline pH, and under the alkaline pH, this BT toxin get activated. And due to the activation of the BT toxin, it started the lysis and inflammation, the lysis and the inflammation of the gut cells, right? So due to which the larva get killed by the starvation. Okay, so the scientists have introduced this gene, this cry gene, in several crop, or we can say in several plants, so that they can protect itself from the action or attack of the pest. Okay. Next is we have the trichoderma species. This trichoderma is a fungus, and they are usually seen in the root ecosystem. Okay. And they are helping to control the plant pathogens. They are used to control the plant pathogen, so that the roots can easily absorb a large amount of nutrient and sufficient amount of nutrient from the soil and help in the growth of the plant. Next we have the baculovirus, right? And especially from the genus of nucleopolyhydro. Okay, they are used to attack the insects and the arthropods. Please keep this thing in mind. These viruses, baculoviruses, are used against the insects and the arthropods, and they are suitable for species specific. They cannot be used in any of the plant. They are very species specific. Narrow spectrum insecticidal application and desirable IPM, that is integrated pest management program, to conserve beneficial insect. These viruses not acting on each and every insect, each and every arthropod. They have their action on the selected species, right? So we have to insert them, or we have to use them on a particular plant and specifically against a particular species. Okay. Now here are some examples for different we can say the agents. So here first we have for the Bacillus thuringiensis. This Bacillus thuringiensis, is used against controlling the black diamond black moth, butterflies, caterpillars on the brassica and the fruit, Helicoverpa on cotton, and the pea pigeon and tomato. So they are using against the several, they are used against the several pests and insects depending on the different species of plant. Now next we have the Bovaria. This Bovaria is used to control the mango hoopers and the mealy bugs, right? And the coffee pot borer. Okay. So, as I said, each and every biological agent has effect on a specific organism. So, these are some examples. Like for the white fly on cotton, we have we are using neem products. The Helicoverpa on gram is controlled by the NVP. The sugarcane borer by the Trichogramma. The roots and the wilt on various crops by the Trichoderma-based products, whereas the fungus Bavaria bassiana is used against the potato beetle and the codling moth. Then we have another fungus which is Antomophytora ignobilis, which is used against the green peach aphid and the potato. That is used against the green peach aphid of the potato. And the last we have the lady beetle and the dragonflies, which are used against the aphids and the mosquitoes, which we have studied initially also. Okay, so here is your question. The question for this concept, and the question is, what does biocontrol refer? Right? What does biocontrol refer to? Your option A is use of biological method for controlling plant disease. Option B says use of chemical methods for controlling plant disease. Option C, use of morphological method by the plants to control the attack of pathogens. Option D says the use of physical methods by the plants to control the attack of pathogen. Okay, so these are the four options. You have to give your answer through this MCQ. So here I am launching your poll. Please respond give your response, or you can feed your answer in this poll. Option A, the use of biological method. Option B, use of chemical method. C, use of morphological method. And option D says the use of physical method. Okay. So, as we know, the name itself says it is a biocontrol method. I'm sorry. It says it is a biocontrol method. That means it is involving the biological method. And yes, you have given the correct answer. The answer is the option A, the use of biological methods for controlling plant diseases. Okay. So now we are moving towards the next, I hope I think the last topic of the day. That is the use of microbes as the biofertilizer. The term says biofertilizer. That means we are using the living organism. We are using the living organisms as a fertilizer to increase the nutrient absorption and yield of a plant, right? So, living organisms. Now, the living organism here in this chapter is all about microorganisms. So here we are using different types of microorganisms as the biofertilizer. That means again, we are not going to use any kind of chemical. Okay. So these are some species. These are some species which are used as the biofertilizer. So the biofertilizer is what? It is an organism that enrich the nutrient quality of the soil. Biofertilizer, it is an organism which enriches the nutrient quality of soil. Why we need to increase the nutrient quality? Because it helps in increasing the yield of the plant, because it helps in increasing the yield of the plant, and it also maintain the health of the plant, right? So in this procedure, or in this method, we use several types of microorganisms. They can be bacteria, fungi, cyanobacteria, right? So first, the most common example which we know is the rhizobium, right? Rhizobium is what? It is a symbiotic relationship. It is a symbiotic relationship in between the root nodules of leguminous plants, or we can say the vascular plants, right? It is, we can say the vascular plants and the bacteria, right? Here we have the symbiotic relationship between the bacteria and the roots of the leguminous plants, right? Why it is present in roots? As you know, the leguminous plants have the high demand for the nitrogen, and that amount of nitrogen is not only be used or absorbed by the roots itself. So the bacteria which is present in the roots of the leguminous plant help them to absorb more amount of nitrogen, and they also fix the atmospheric nitrogen which is present around the plant into the nitrates and nitrites, so that the plant can get a good amount of nitrogen supply. Okay. Then we have the free living bacteria in soil, that is azospirillum and azotobacter. They also make soil nitrogen rich. Now the next is mycorrhiza. Firstly, we have done the rhizobium. Now we have the mycorrhiza. Now in rhizobium, we have the symbiotic association with the bacteria, right? But in mycorrhiza, we have the symbiotic association with the fungi, that is fungus. Okay. So fungus has the symbiotic association with the roots of the vascular plants, and the example of this fungus is glomus. What these fungus are performing here, they absorb phosphorus from the soil and pass it to the plant. Please keep this thing in mind. The bacteria which we have used, were helping in fixing nitrogen, whereas the fungi is helping for fixing phosphorus. It is helping in fixing phosphorus. Okay. Next, they give resistance to root-borne pathogen. As the fungi is already present there, so they are not allowing any of the pathogen to start living there or start destroying the roots. They also tolerant to salinity and the drought. Drought is the condition where the water scarcity is present, and salinity, that means the salt concentration is high. So in salinity, that is salt is high. In drought, that means the water level is low. So in both the conditions, these fungi are able to survive. So by the help of absorption of good amount of phosphorus and maintaining the plant resistant and tolerant to the pathogens as well as to the climatic condition, they are helping in the overall growth of the plant and the development of the plant. Right. Next, we are moving towards the cyanobacteria. In cyanobacteria, we have the blue-green algae. You can see the filamentous rod-like structures here, which are present in this picture. These are the cyanobacteria, that is blue-green algae specifically. So they are autotrophic microbes. Autotrophics, that means they make their food on themselves, and they again help in the fixing of nitrogen. They are again helping in the fixing of nitrogen. The example of cyanobacteria is Anabena, nostoc, and oscillatoria. They are basically found in the paddy fields. Paddy, that means rice field. The rice field had a good coagulation of water. The fields are they are having a good amount of water supply. The water is kept filled in the fields so that the paddy can grow. So the cyanobacteria serving as a biofertilizer, and they also add the organic matter to the soil to increase the fertility of the plant. Okay, so here is a question related to this concept or this topic. The question itself the question says, which of the following statement is not related to the mycorrhiza? Please pay attention. We have to find out the statement which is not related. Here we have to talk about the statement which is not related to the mycorrhiza. So your first option says the many members of the genus Glomus forms the mycorrhiza. Okay. Your option 2 says fungal symbiont absorbs nitrogen. C, the plant show resistance to root-borne pathogen. And D says there is an overall increase in plant growth and the development. Okay. So here we have the 4 different statements. Now you have to find out the statement which is incorrect regarding the mycorrhiza. And here is your poll. You can give your answer through this poll. You have to find out the statement which is incorrect regarding the mycorrhiza. Okay, so as everyone has given their answer, I am closing the poll. So this is your result. As you can see, everyone has opted for option B. Right, you are right. We have already talked about the biocontrol, right? In a biofertilizer, the family for the fungus is basically Glomus, right? They are all belonging to the genus Glomus. That means this point is correct. The point C says the plant show resistance to the plant-borne pathogen, right? Root-borne pathogen. If the fungus is already present there, it will not allow another pathogen to stay in the root, or they can attack the root. So this point is also correct. Due to the presence of fungus, the root-borne pathogen are not attacking in the plant. Then we have the option D, which was saying there is an overall increase in the plant growth and development, right? Because it is helping in the absorption of the nutrients from the soil. It is also making our plant the resistant and tolerant to pathogen as well as to the adverse climatic condition. So in these points, the plant is getting the overall development. So this third point, the fourth D point is also correct. But the B point says the fungal symbiotic absorbs the nitrogen. No, nitrogen is not there. These are responsible for the absorption of phosphorus. They are absorption of phosphorus. The nitrogen is absorbed by the bacteria. It is fixed by the bacteria. That is why this statement becomes wrong. And as this statement is incorrect, that means it is the correct answer for our question. Okay. So this is all for today. This is what we have to do today. Okay. Now, thank you so much. If you have any doubt, you can leave your doubt. You can ask your doubt in the chat box, or you can leave your doubts in the chat box so that we can resolve your doubts in the next class. Okay. So before leaving the class, please give your feedback through this NPS poll. Right. I'm launching the NPS poll. And after giving your feedback, your precious feedback, you can leave the class. Okay. Okay, so thank you so much for your feedback. Okay, we'll meet tomorrow with the new topics, or maybe we are going to practice some new previous year questions, right? So tomorrow onwards, we are starting the practices of our previous year question. So this is all for today. We'll meet tomorrow with a new topic. Okay, have a great evening and a good night, students.